He said that when the cell is doing its job, when it's in G1, S, and G2, when the cell is just doing its job, its DNA is unwound, and it's in the form of what we call chromatin. Imagine the difference between having, 24 balls of yarn stacked up in neat little rows on the floor versus taking those 24 balls of yarn and unwinding each one of them into just a big giant pile in a bucket. All right? In the big giant pile in the bucket, we call that chromatin. When we take and we wind them up into neat, easy to see bundles, that's called a chromosome. So during this prophase, that's when we're winding up. That's when we're taking each strand out of the bucket and we're turning it back into an easy to recognize ball of yarn. We're turning it into this structure that we recognize as a chromosome, those very recognizable X shape. It's going to twist, 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 just like you wrap up a ball of yarn. We're going to wrap up the chromatin. We're going to wrap up the DNA into this X shape. Not exactly as easy as a ball of yarn, but that's how it's done. While the chromosomes are condensing, they're going to start to line up around the equator of the cell. If you can imagine that the cell is very similar to the world, what happens is the chromosomes will line up along the equator, the center of the cell. During this time, if it is an animal cell, what we call the spindle apparatus begins to form. So as my chromosomes are lining up, they line up along the equator of the cell. Before we begin mitosis, the first step during prophase, the cell is actually going to dismantle the cytoskeleton. Remember those microtubules and intermediate filaments and stuff like that? The cell is going to dismantle all of that and then reuse it, reconstruct it into this structure called the spindle apparatus. The spindle apparatus is formed around those centrioles that we talked about in chapter, 4? Those centrioles are at the top and the bottom of the cell. And then what happens is those fibers, the microfilaments, will go down and extend to that belt buckle. One fiber will come from the top of the cell and attach to the belt buckle, and another fiber will come from the bottom of the cell and attach to that belt buckle. And so all of my chromosomes get a wire that comes from the top and another wire that comes from the bottom and attached to my chromosomes. All right? That's what happens during metaphase. Meta stands for middle. It's not the middle of the process. It's called the middle because the chromosomes line up in the middle. Here we see interphase. Interphase is when the cell is just doing its job. During interphase, at the end of interphase, it's going to enter prophase. And during prophase, what happens is this nuclear envelope disappears. The cell skeleton is deconstructed and reconstructed into the structure called the spindle apparatus. As we progress from prophase towards metaphase, we can now see that the chromosomes have been made. They've wound up. Most of them have wound up. Some are still winding up. And some wires have begun to attach from the bottom and the top of the cell down to each of these chromosomes. Here's one chromosome right here. There's a wire coming from the top, and another wire coming from the bottom. And notice how they attach on either side of that. They're actually attaching to one copy of that double chromosome. Remember that each chromosome is not one. Each chromosome is actually two copies of the same. After metaphase, after that spindle apparatus forms and wires have attached to all of the chromosomes, now it's time to separate them. And the process of separating is called anaphase. Ana means away or apart. So this is the phase where the chromosomes get pulled apart. The cell physically stretches. The cell physically gets longer. That spindle apparatus, part of it is that those microtubules will push past each other. And when they do, they pull those ropes, those wires, tight. When they pull those wires tight, they physically rip the chromosomes in half. The chromosome can't stand up to that kind of pull, and so one gets pulled to the top, and the other one gets pulled to the bottom. All right. I notice, and you notice, that they're bending backwards now. It's because they're not rigid structures. They're flexible. They're soft. Like if you pick up a rope in the center, it's going to bend. They flop because they're not rigid. They're soft. Chromosomes are ripped in half and drugged with the poles in anaphase. In telophase, the chromosomes will decondense. They begin to unravel. The whole point of raveling them up into that nice chromosome shape is so that I could separate them easily. Well, what just happened? In anaphase, well, I just separated them. Once they get where they're going to the ends of the cell, it's now time to unwind them. The only reason why I wound them up is to make them easy to separate. Well, I've separated them, so I'll unwind them. Telo means ending or finished. So telophase, the ending phase, is it's time to start turning these this one cell, it's time to start turning it into two. The chromosomes will unravel, that nuclear envelope will begin to reform, and the chromosomes, as I said, they will begin to go back into that chromatin phase. The cell itself, though, hasn't finished the dividing process. That takes place after the end of mitosis, and it's called cytokinesis. Cytokinesis, or sometimes called cell division, is when the one cell is physically split in two. Here are my chromosomes that are lined up along the equator of my cell. Those, the spindle apparatus, those centrioles have organized the spindle apparatus so that where the wires are coming down to each chromosome. You can't really see so well, it's kind of hard to see. You can see it better in this one. These are those microtubules that are slide past each other to stretch the cell out. You can kind of see them if you look really close. There's one right there. There's another one right here. When the cell begins to stretch, those microtubules slide past each other. The ropes themselves, the wires themselves get shorter. They get pulled back down again. And now my chromosomes get torn, and they're getting pulled to the poles. At the end of anaphase, I'm now at telophase, and notice how the chromosomes are starting to unwind. The whole purpose of having them in this shape was to make them easy to divide. They've been divided. So they start to unwind. The cleavage furrow begins to form. The cell begins to split in two, but what hasn't happened yet? And the answer is, it's still technically one cell. It still isn't split in two. The splitting in two is this very final step called cytokinesis. I start with one cell. It lives its life. Towards the end of its life, it's going to enter this process called called mitosis. Prophase is when that nuclear envelope disappears and the chromosomes begin to condense. These condensing chromosomes begin to line up along the equator. When they do, during this process, that spindle apparatus is formed. The spindle apparatus wires attached to each chromosome. Anaphase, the cell stretches, and when it stretches, it rips apart those chromosomes, and it pulls the chromosomes to the poles. Now that the chromosomes have been divided, it's time for them to unravel, and they begin to unravel. The nuclear envelope begins to reform, but we're not quite done with this process of division yet. Finishing the process is called cytokinesis. That cytokinesis is not part of mitosis. It is part of cell division, but it's not part of mitosis. Once my cell has physically become two, and now I have two cells that are going to live their lives. This is the process of mitosis.